Later, I will introduce our lab member after your talk. So as time comes up, let's start. So let me introduce today's speaker, Andrew Phillips. So Andrew Phillips got the PhD at physics in University of Sydney. And then he did a postdoctor at the Harvard Medical School. And then he came back to Australia to Monash University. And then he's leading this he's a really world expert in circadian rhythm, human circadian rhythm and sleep. And he using the mathematical model and statistical analysis to understand this complex phenomenon. And he will talk about this today. And let's welcome today's speaker.
Thank you so much for the introduction. I hope you're all doing well there. It's good to see some familiar faces. I really had a very fun time last time I was visiting your group. It's unfortunate no one's able to travel at the moment, but hoping to get back there as soon as it's possible. So what I wanted to talk about today was some of the recent work we've been doing in basically trying to individualize predictions of the human circadian system. So this is just sort of a quick summary of the main areas I'm working at the moment. So my research group I've sort of assembled at Monash now. Here are some of the key members. And there are kind of three main areas we're working in. One is understanding the impacts of light on the circadian clock. And some of that has come out of sort of experimental work, looking at the sensitivity of the human circadian system to light recently. Another area I'm looking at is sleep regularity. So that's something I started in 2017, and it's become sort of about a third of my research program now, just because we're finding that sleep regularity is a really useful predictor for many different health outcomes, and potentially a good target for interventions. And then the third, which is probably the most salient today, is the mathematical modeling. And a lot of the work I'm going to be presenting today, one of the key players in that has been Julia Stone, who's my current postdoc at Monash. So I'll reference some of her work as I go. So the sort of problem I want to talk about is, how do we measure circadian timing accurately and cost efficiently in real time? Or in other words, is there some way that I could just, know what is my internal circadian timing or your internal circadian timing very easily at that particular moment? And the answer, I hope, is models. I think mathematical modeling has a lot of promise for answering this particular question. It's a cost-efficient approach, and it potentially can make predictions in real time, which other methods can't. I probably don't need to tell any of you, but our bodies are made of many trillions of clocks. And we know now that they're essential to our health. We know these clocks are all through the body. We know that there's a central circadian clock that regulates those rhythms. And we make a pretty big deal of this Nobel Prize that was awarded a few years ago. We put it in every grant proposal. We reference it in our reviews. It's this key kind of issue of pride, really, for the field. And yet, it's if you look at it compared to other Nobel Prizes in medicine and physiology, it's one of the few areas of breakthrough that hasn't really had any clinical translation. You don't get routine phase assessments. You don't get circadian assessments in medicine. You don't go and see your personal physician and get any form of circadian measure. The only place where we even really do that is in people with circadian rhythm disorders. So it's not mainstream medicine, and we think it should be, right? We think this clock is essential to so many areas of health. So how do we do that? I think there are many reasons we would want to be able to accurately measure the circadian clock. Those are both clinical and non-clinical. One would be correct timing of circadian interventions, things like light therapy or drugs that have different effects at different circadian phases, so the idea of chronotherapy. Also, understanding, diagnosing, and treating disease. Like, we know that circadian disruption is probably a key part of the development of many disorders, including mental health disorders and including metabolic disorders chiefly. So, being able to measure that in someone might be a useful way of understanding the treatment of their disorder. And, that could be totally aside from someone with what we would consider a circadian rhythm sleep disorder. That could be someone with diabetes. Also for predicting functional outcomes, when we're talking about people's performance in the field, or when we're looking at shift work or optimal scheduling of sleep, all of that relies on accurate prediction of circadian timing, because as we know, the circadian clock regulates when we're most alert and when we're most sleepy across the day. Unfortunately, there are current barriers to actually measuring circadian timing. The measures that we have are logistically challenging, so they're not that easy for people to actually undertake. They often have to be done in a controlled environment, or you have to give people very careful instructions for how to do it at home. It tends to be quite costly, and we also don't tend to get the results until weeks later. And that's not great. Like, for example, if we're studying a shift worker and they're on a rotating shift schedule, by the time we actually have their measurement weeks later, they might be at a totally different circadian time. So now the measure is essentially irrelevant. The gold standard we use in the field is melatonin. Formerly in the lab, temperature was sort of the gold standard for circadian rhythms. Unfortunately, temperature is very easily masked. You go for a walk, or you have a shower, or you eat a meal. All of these things, modify your body temperature rhythm, and so it's actually virtually impossible to demask that signal and to get to the endogenous circadian rhythm from temperature in field conditions. You can only really use it under very controlled conditions in the lab. And so melatonin has sort of become the gold standard in the field, but it also has limitations. It has to be collected under very dim conditions. We know that it really doesn't take very much light to suppress the level of melatonin. And it also requires controlled posture. And it also requires people to have a controlled diet during the period of collection. So that's not particularly easy for people to do. And as I've mentioned, those results can take weeks to be known, because then the samples have to be sent for assay to some lab, and then you get the results back. And sometimes that will even take months. So it's not something that we get back very quickly in a clinical context. You're probably aware there are other biomarkers being developed. So people are really keen to develop, there's work going on at SARI, there's work going on at Monash, there's work going on in the Boulder group, all looking at ways of, could you take like a single, biological sample, whether that's blood or saliva, and by taking lots and lots of, for example, omic measures, could you get some sort of estimate of circadian timing that way? But right now, those methods have only been used under basically lab conditions like constant routine. They're not really well validated for field use. And they remain prohibitively expensive. They're not something that's going to be mainstream medicine for, as optimistic as we always are, I would still not be holding my breath on that one. I think it could be decades before those are actually useful. So, I think there's this long period before any of those methods are going to work where we still want to be accurately predicting individual timing. And that's where models might be fit for purpose. So, this alternative approach is to use mathematical models or statistical models. As we know, light is the key input to the circadian clock that it determines its timing in humans. We know this from lab studies. And so these types of methods, we can use inputs such as light exposure, and also potentially use other physiological measures from biosensors like activity or temperature to predict someone's circadian timing. What I'm going to focus on is this particular mathematical model. Here's just an overview of it. I'll go into a bit more detail in a moment. But this is a model that was developed at the Harvard group of the human circadian clock based on the many studies that they ran in their lab there. It's basically a limit cycle oscillator model. So it represents the fact that our internal circadian clock is a self-sustaining oscillator, and it's an oscillator that can be shifted by environmental stimuli, with light being the most impactful environmental stimulus. So it has what's called a phase response curve, meaning that its phase can be shifted either forward or backward, depending on the time in the cycle that an individual gets light exposure. So that's the basic idea of this model. So let me just go into a little bit more background on that model for those who aren't really that familiar with it. So if you imagine we have a system that has a limit cycle, so it is a system that will continue to oscillate. It has a preferred frequency and amplitude. We might perturb that system, for example, by giving it a light exposure at a particular time. And if we wait a while, the system's going to find its way back onto the limit cycle. But where it ends up back on the limit cycle might be a bit different from where it would have been had we not perturbed it. And so we've caused a phase shift. That's that delta phi that I've put there. There is this sort of concept of isochrons, which are points that will ultimately end up at the same point on the limit cycle. So you can imagine there are many different ways in which I could perturb the system. And some of those perturbations will lead the system back onto the limit cycle at the same point. That means we can kind of recognize those points as being at the same phase, even though they're not sitting on the limit cycle initially. So that's the idea of an isochron. And so this is what we mean when we're talking about phase shifting. This kind of system, there's some perturbation, And then when the system finds its way back onto the limit cycle, it's now shifted compared to where it would have been, either further forward or further back, depending whether it's a phase advance or a phase delay. We know from a bunch of physiological experiments that humans have this phase response curve for light. So that's what's plotted down below. So this is circadian phase on the x-axis. So that's where in the circadian cycle the stimulus is applied. And on the y-axis, you have the size of the phase shift. So 0 would mean no shift. A positive value means an advance. So you've pushed it further ahead in its cycle than it would have been. And a negative value means a delay. So it's been pushed further back behind where it was initially. So you can see for the human circadian clock, this is the phase response curve for a bright light pulse of 6.5 hours. And what you can see is that you can get an advance up to about 2 hours. So the highest y-axis value is about 2. And you can get a delay of up to about 3 hours using this type of light stimulus. And where the system tends to be the most sensitive is centered around the biological night. So you get the biggest phase delays sort of in the late evening, after the sun has gone down, into when you would normally be sleeping. And you get the largest phase advances sort of in the, just before dawn and into the early morning. And so this type of response has been put into this mathematical model to represent how the system responds to light. Here's sort of my latest attempt. I don't know, every talk I give on this model, I try to come up with some different diagram that helps. So this is my latest attempt. So these are the different components of the model. So over the left, there's this very simple model of photoreceptors in the retina, which includes the activation of the photoreceptors and their recovery during darkness. And that's represented by a first order ordinary differential equation with the variable n. So n is the sort of fraction of the photoreceptors that are being activated. And the input to that is alpha, which is a function of I. I is the light intensity. So basically, light drives what happens to the photoreceptors in this model. This then generates an output called B hat, which is then modified to become B, and that is the photic drive or the light input to the circadian pacemaker. And then on the right, you have the circadian pacemaker itself, which is a limit cycle oscillator. And what it is it's a modified Van der Pol oscillator with all the parameters having just been set to sort of match what's seen in human physiology. So this model was sort of fit over many years to different lab experiments, where they tried exposing people to different patterns of light, or they tried suppressing their amplitude or shifting their phase with different patterns of light. And they just looked at how the system responded, both to the perturbation itself and in its recovery following a perturbation as it came back onto the limit cycle. So we know that this model is a pretty good representation of sort of average human healthy physiology. That's been pretty well established at this point. What we don't really know, or what we didn't know until a few years ago, is how the model performs outside of those settings. Because, firstly, the lab is a kind of artificial environment, and the types of light exposures that we see in the lab are quite different to those in the real world. If you've ever done any light recording in the real world, you'll know that the types of signals are very noisy. You'll see it's a very spiky signal. Someone will go for a walk outside and all of a sudden they're up at 10,000 lux. The next minute they come inside, they're back down at 100 lux. It's actually quite a dynamic signal. Whereas when we do these types of lab experiments, the light is like a square wave. The light is on at a particular level and constant, and people are even controlled in where they're looking in the room usually, so that we have a very constant, well-controlled level of light going into the eye. And then the light is off and it's flat at 0 lux, and there's no intermediate setting. So we don't really know how well the model would translate actually to to field conditions, considering that. The other thing is that the people who get screened in the Harvard Sleep Lab are extremely homogeneous. They're very, very healthy sleepers. For a lot of these studies, maybe like 3% of the participant applicants will actually get into the study. Most will be excluded on various criteria. So the people who get in, are not necessarily representative of general physiology. That they don't really often represent the extremes. There will be no very early sleepers or very late sleepers within that group. So it is a highly homogeneous sample. So what we wanted to do was, see how well this model would perform out in the field. And we attempted in 3 settings. One was a study I did a few years ago comparing highly regular sleepers to highly irregular sleepers. A second study, which was completed as part of Julia Stone's PhD, was in rotating shift workers. And then a third study was completed by another PhD student, Jade Murray, who I co-supervised, who was looking at individuals with delayed sleep-wake phase disorder. So looking whether we could actually predict phase in a clinical population. So in study one, basically what we did is we studied 60 college undergraduates for a couple of weeks. And then we computed which individuals had the most regular or the most irregular schedules. And we took the 12 most regular and the 12 most irregular. And in those individuals, we measured their circadian timing using the gold standard dim light melatonin onset. And you'll see what we found in general was that the people who had regular sleep timing were a couple of hours earlier in their circadian timing than the individuals who had irregular schedules. And so what I wanted to understand with the model was, could I predict this group level difference between them just based on their light exposure patterns? So can I explain the difference between regular and irregular circadian timing just based on the differences in light that they get? That's sort of a null hypothesis. An alternative would be that they have some physiological difference, and that's why they're different in their circadian timing. But I wanted to test this null hypothesis of, could it just be their difference in light exposure patterns that accounts for this average group difference? And what we found was two things. Firstly, that yes, you can explain almost all of the group level difference just based on the individual's light exposure patterns. So this is with the actual DILMO on the x-axis, and this is the model predicted DILMO on the y-axis. And you'll see if we look at the group difference in means, the actual difference is 2.2 hours. The model predicted difference is 1.7 hours. So at the group level, the model is actually accounting for that difference. What you'll also notice, though, is this is a bit of a sneeze, looking at the plot, like the points are scattered all over the place. And what that's telling us is that at the individual level, the model's not doing great. Neither in the regular nor the irregular group is the model doing a great job of predicting an individual circadian timing, which we perhaps should expect. That given that the model was trained on group level data, it was never really tested on individual level data. It would be kind of remarkable if it did work there. So that was the first finding using this model in the field, which was satisfying on one level, but also opened this new question of, well, why is the model not doing so great at the individual level, and can we do better? The second study was in shift workers, and shift workers are a useful group to study because their circadian rhythms are just, chaos, really. Like they're, you almost couldn't design a worse condition for a human circadian system than rotating shift work. It's reminiscent of some of the lab studies we actually designed for animals to induce circadian disruption. And so, I often see this as a useful setting for model testing because it's an extremely stringent test of a model. If a model can accurately predict timing under these conditions, it's likely to work well in other conditions. And one thing you'll note if you go through the literature on sort of circadian phase prediction methods is many of them have really only been validated in people who are on regular schedules, who are living on a day schedule. And that's a relatively soft case. If you can get that done, it doesn't necessarily translate to shift work. So, shift work is a really interesting test for the model. And so what Julia looked at here was, basically medical staff who were transitioning between day and night schedules. So had both a diurnal schedule assessment and a nocturnal schedule assessment. And in this case, circadian phase was assessed using this urinary marker of melatonin AMT6S. And light was wrist one recordings for 1 to 2 weeks, similar to the first study. So what was found in this one, again, was that at the group average level, the model's not too bad. But when you get to the individual level, you'll see in the diurnal schedule, the mean prediction error was about 0.7 hours. When you get to the night schedule, which is, of course, the more variable condition, you get an average error of about 1 hour. That is the absolute error. So in this case, what you'll notice, so the black points, sorry, I should have just described, are the actual data. The squares, so the open symbols are basically two versions of the model trying to predict circadian phase. And what you'll notice is in general, near the middle of the plot, so that is for the individuals who have sort of the average timing or close to the average timing, the model does actually very well. It's when you get out to the extremes, like the far right end of the graph or the far left end of the graph, that you start to see these big mismatches between the model predicted timing and the individual's actual timing, such that some of the errors, might be 2, 3, 4 hours for an individual. And for clinical applications, that's really not good enough. We know that we need to be able to predict more accurately than that. For most sort of applications where you would want to know someone's circadian timing, for example, for delivering a drug or delivering a light intervention, we definitely want to be within about an hour. Anything bigger than that can lead to big problems. For example, if you think back to the phase response curve, if you're off by 2 hours, that can be the difference between being at the peak phase advance point and being at almost no phase shift. And if you're off by 4 hours, what you thought would be a phase advance might actually be a phase delay. So you've got to get it right to a greater level of accuracy than that. And so again, this is kind of what we might have anticipated for a model that's been trained at the group average level. It does well for individuals who sit close to the average, but for individuals who are at more extreme phenotypes, it's not doing so well. The third study was looking at individuals with delayed sleep-wake phase disorder. So I thought this was also an interesting test case because the model was only ever trained in healthy individuals. And so we wanted to see how it would perform in this fairly large group of 154 individuals with delayed sleep-wake phase disorder, where they had their salivary dim light melatonin onset assessed once, and we had wrist-worn recordings of light for 1 week. What I'll note is that when we first tried the model with just default parameters, it kind of did what you would expect. It predicted phase too early for these individuals systematically on average. And the average phase prediction error was, I think, around about 1.7 hours. What we did with this one when we allowed some of the model's parameters to be trained, basically on half of the data set, and then make predictions on the other half of the test data set, we found that the mean absolute prediction error was similar to what we were finding in shift workers before, around about 1.1 hours in this case. And again, we've got the same phenomenon. Like if you look at the error between the model predictions and the individual, it does pretty well near the middle of the range. But if you go to the extreme right or the extreme left, again, you've got larger mismatches. So the model's systematically mispredicting either too early for the individuals who are late or too late for the individuals who are very early. And this paper just came out in scientific reports, by the way. The last 2 papers were also in scientific reports. So apparently, if you want to publish circadian phase prediction, you should go to scientific reports. That's my recommendation.
Andrew, quick question.
Yep.
So here you train the parameter with this training data set. So you change the parameter.
We did. So we allowed 2 of the parameters to vary. One was tau. Because there is one paper showing, or at least claiming, that individuals with delayed sleep phase disorder have a longer tau than healthy individuals. And the other was a light sensitivity parameter that basically made the phase response curve more delay heavy than advance heavy, because we recently found that individuals with delayed sleep have a bigger phase delay when given the same light pulse at that time of day. So I thought those were the 2 sort of most plausible mechanisms for explaining the systematic difference in the model. Yeah. But if we just throw the default model at it, it does worse than that. The error is like 1.7 hours. So it's clearly off. And so I would take this as sort of evidence that we need to do better with the model parameters if we're ever going to get things right at the individual level.
Thank you. Got it. Thank you.
Yeah, no worries. So, okay, let's think about, possible reasons for inaccuracy at the individual level if we step back. One, the model could just be wrong. And that would be bad. We'd be back to the drawing board. I don't think that's true for a number of reasons. I think this model has been very carefully developed over many years. I think it's pretty well validated and it has a strong physiological basis. So I don't think the model structure is just off. I think that's an unlikely answer. Wrong parameter values could certainly be part of it. I just told you, in the delayed sleep phase disorder, that we had to adapt the model parameters even to get the group average level estimates right. So that tells you that there are individual differences, because if we move into different subpopulations, we do have to adjust some of the parameters. And, there are potentially individual differences in physiology that would explain why the errors are greatest for those individuals who have the most extreme phenotypes, because you would expect those to be the individuals who have the most extreme deviation from average model parameters. So, deviating from default parameters. Another possible problem could be wrong inputs to the model. We may be inaccurately measuring the light input, because all of these studies are based on wrist-based assessment of light. And we know our eyes are not in our wrist, and what we're capturing at the wrist is probably not exactly right for the eye. And the way in which we measure light is also not ideal. We tend to measure photopic illuminance, which is visual brightness. And we know now, based on the physiology, that that's not really how light is processed by the circadian system. So there's a couple of ways in which we might be able to improve that input, and I'm going to talk about that a bit later in this presentation. I'll also note something that I think people often overlook is there are also fundamental limits on accuracy just based on temporal resolution of the phase marker. When we do melatonin assessments, like salivary assessments, typically our resolution is 60 minutes. We tend to get one sample every 60 minutes. So theoretically, you can't do a whole lot better than that. If you get your error down to 30 minutes, you might already be at the limit of what's actually achievable given the resolution of the phase marker itself. So in terms of potential sources of individual variability at the parameter level, there are a couple that we decided to explore. One is circadian period. This is not a new idea. This has been previously modeled to account for individual differences. And we know, even in individuals who went through the Harvard lab studies and were highly homogeneous in their sleep, that there can be big individual differences in intrinsic circadian period. That's what that histogram is showing. It's a group of 157 individuals who all went through forced desynchrony studies. And you can see that the intrinsic period, even in these individuals who are all intermediate types, all super healthy sleepers, ranged from about 23.5 to about 24.7 hours. One of the problems with tau, of course, is it's very difficult to measure. You basically have to do a forced desynchrony to assess someone's circadian period, at least using, what we know today. The other thing that sort of cropped up for us recently is light sensitivity. So I was asked to come and help look at this data set that had been collected, where basically they were looking at the dose response curve to light in a pretty good sample of individuals. There's almost 60 people. And it was an unusual study in that each of the people had come into the lab 6 or 7 times to get a light exposure at a different level, allowing you to actually map individual level dose response curves for the first time. And what sort of stood out when we first looked at the data set was that it appeared very noisy. There was a huge amount of variation in the data set. But what I found was that, a lot of that noise was actually into individual differences. And when you went down to fitting dose response curves at the individual level, you tended to fit quite well. The dose response curves were actually fitting the data quite excellently at the individual level. It's just that there were these huge differences between individuals in sensitivity, which were kind of unexpected. I mean, certainly I expected there to be some individual differences. I didn't expect them to be like 50-fold. And that's what we found. We found that for like the least sensitive individuals, you needed to get them over 400 lux to suppress half of their melatonin. Whereas for the most sensitive individuals, already at 10 lux, we were suppressing more than half of their melatonin. And in terms of real world lighting, that has huge implications. We spend like 80% of our time indoors these days. And so we spend heaps of time in this intermediate dynamic range for the system, where these individuals' differences could make a huge difference, right? If you think about this person who is highly sensitive and only requires 10 lux to get a meaningful physiological response, that means all indoor light for them is essentially daylight. Like it's having this huge impact on the system. Whereas for this very insensitive individual, you need to get them up to very bright indoor light or even sunlight to actually have this strong effect, at least on melatonin suppression. And this hadn't been explored previously as a potential basis for individual differences in circadian timing in a modeling context. So it was something we wanted to look at and see, could this potentially account for some of the differences that we see? So that was sort of the question we asked, could we improve individual level phase prediction by adjusting light sensitivity parameters in the model? And is that comparable to adjusting intrinsic circadian period? Because the intrinsic circadian period has always sort of been the default choice for explaining individual differences. We always talk about, someone who's a late type probably has a long tau, someone who's an early type probably has a short tau. But I began to wonder, based on these data, whether some of these light sensitivity parameters might actually be just as important. And so we collected this sort of pilot data set. It was in 12 healthy individuals. We had 3 weeks of continuous light monitoring. And what was novel about this study is that we had 3 circadian phase assessments in every individual. So what that meant was we were able to look at intra-individual variation in circadian timing, as well as inter-individual differences. And what we attempted to do was look at the model's sensitivity to 3 of the key parameters and see if we could, by fitting those parameters, improve the estimates of delmo. And so this is sort of a proof of concept, I guess I would say. This is not a prospective prediction. This is just saying, can we do better in terms of model accuracy by potentially adjusting some of these parameters? Because if the answer is yes, then that leads us next to saying, well, okay, could we take some sort of physiological assessment in someone to get an estimate of one of these parameters and plug that into the model? Because if we can do that, if it just requires, say, like a 10-minute pupil area assessment or something, and that allows us to estimate a model parameter, then that's fantastic. We might now be able to individualize model predictions in a very easy, straightforward way. So the model parameters we investigated, just sort of going back to that diagram of the model, one was this parameter P, which basically affects the shape of the dose response curve for the model. If you take a lower value of P, you get a more sensitive dose response curve in the model, or a sort of lower ED50, if you want to think of it that way. Another we looked at was K, which is this parameter that affects the phase delay bias in the model. Basically, is it more phase advance favored or more phase delay favored? And then tau, of course, again, the intrinsic circadian period, which is a parameter most people have looked at varying in this type of context. So just to sort of show you the effects of varying those two parameters, P and K, on the model, what I've done here is simulated basically a phase shift experiment using the model, and then looked for different levels of photopic illuminance, what would be the predicted phase delay. You can see the black line is sort of the model's default prediction. So you've got this nice dose response curve up to 10,000 lux. You can see if I make P smaller, if I bring it down to 0.2, I get a more sensitive response. The phase response is more sensitive at lower light levels. I can get quite a big response at 100 or even 10 lux. Whereas if I make P bigger, you can see you get this sort of later phase response where you need to get up, more like our really insensitive individual that we saw in that study who required over 400 lux to get to 50% melatonin suppression. And then on the right, you can see the effects of varying this parameter K on the phase response curve. You can see the default is that black curve, which is similar to what I showed you earlier from the actual data, where you've got a phase advance region, a phase delay region. The phase delay region is a little bigger than the phase advance region. And if you change the value of that parameter, you can either make the phase delay region bigger, that's like a bigger K, or make it smaller and favor the advance region. That's the blue curve. So that's sort of just showing what the inputs are. So the first thing we did was a sensitivity analysis. So for each of these panels, I've got one of the model parameters on the x-axis. So either P, K, or tau in each of the panels. And on the y-axis, I've got the predicted Dilmo time. There are lots of different colored points because each color represents one of the participants. So what this is showing is, what would be their average predicted Dilmo time based on varying one of the model parameters, but taking their actual recorded light data as the input to the model. And so you can see that the two panels on the right are pretty straightforward to interpret. If I make K bigger, that is, if I make the phase response curve more delay biased, then predicted Dilmo goes later. And if I move K more negative, that makes the phase response curve more advance favored, and that pushes circadian phase earlier. And those are just monotonic responses, exactly what you would predict. The far right panel is also exactly what you would predict. Longer tau means later Dilmo in the model for the same light input. And that's also a monotonic response. On the left side, this was a little bit more of a subtle response, changing the dose response curve. You'll see there isn't actually much systematic change across the range from left to right. Changing P over its full range doesn't really have much effect on the average phase. But if you look at the individual curves, it's interesting. The response is not monotonic. Some individuals, as you decrease light sensitivity, become later. And some individuals, as you decrease light sensitivity, become earlier. And you can see that by tracking different colored points. Some of them shift earlier, some of them shift later across the x-axis range. And we were able to sort of understand this by looking at like a really sort of simplified case. Like if you imagine an individual with higher evening light, so you can see, imagine that they've got sort of, relatively low light levels throughout the daytime, and then they've got this very high evening light level. So these are, of course, kind of extreme cases just to demonstrate what's going on. And then you've got an individual who gets actually most of their light exposure in the morning on the right. Then we can look at how that translates the inputs into the model. In those middle panels, we're looking at the photic drive into the model. And you'll see for the individual who has high sensitivity, both the daytime light and the evening light are enough to activate the system because it has this very highly sensitive dose response curve. The difference in the light levels is not so important because we're near saturation even at the lower light level. Whereas when you've got lower light sensitivity, the model kind of isn't affected by that lower light level early in the day, and it is affected by the light level later in the day. Whereas if we look at the example on the right, it's the other way around. That it's the early morning light that's brighter that has an impact, and then the evening light has basically no effect for the low sensitivity version of the model. Whereas for the high sensitivity version of the model, it's similarly affected by both of those light levels. And so finally, if you look down at the bottom panels, you can see that the same variation in the parameter value can lead either to an earlier phase with high sensitivity or a later phase with high sensitivity. You can see the blue curve is either shifted earlier or shifted later than the red curve, depending on the light input that I've given to the model for the same parameter values. So in other words, there is an interaction between the model's light sensitivity and the light input that I give to the model that determines the circadian timing. It's not a simple matter of saying, if you have high sensitivity, you'll be later or earlier. I need to know your light sensitivity in combination with your light input pattern to determine whether your phase will be earlier or later. So, if you're an individual who is high sensitivity and you also tend to use a bunch of light in the evening, then yeah, you're probably going to be someone who gets shifted later. But it's not just uniformly the case that high sensitivity means later phase. It's dependent on how you use light across the day. What we then looked at is, whether we could improve the model's default predictions by optimizing these parameters on an individual level. And of course, because we had 3 DILMA assessments in each individual, the system was identifiable up to 3 model parameter fits. So we looked at P, K, and tau. You can see if you fit all of those, you do extremely well. That's in panel B. But what was interesting was that, even just fitting both of the light sensitivity parameters, so P and K, that's up in panel E, you can see that the model prediction accuracy is much better than the default. So the story from this is really that, tau is overall the most important of these 3 parameters for accounting for individual differences. But you can account for almost the same amount of individual variation using light sensitivity parameters in the model, which doesn't tell us that light sensitivity parameters are responsible for individual differences, but it tells us that it is an equally plausible mechanism. And it's one that's now worth exploring. If we start to take light sensitivity assessments and put them into the model, can we begin to do better on individual level prediction? So I would say as a proof of concept, it was successful. Okay, so now what I want to tell you about, maybe I'll just ask, are there any sort of immediate questions on the modeling before I talk a bit about light? But I'm happy to also take a bunch of questions at the end if you want me to keep going.
Andrew, I think you can keep going. Yes.
Okay, sounds good. I'll try to finish a bit early, so we've got a bit of time. Great. Okay, so the next thing I wanted to talk about is, how we measure light. I mentioned before that, how we measure light using wrist-worn sensors is kind of a flawed approach. And I say that having run hundreds of participants with wrist-worn sensors myself. But, if we think about it, we've got our light sensor on the wrist. And there's a few problems with this. One, of course, wrist-worn devices are prone to coverage. If you've ever run a study with wrist-worn sensors, you get all these periods of like 1 lux or less during the daytime that clearly weren't really what the person was exposed to, that you end up having to clean out or impute somehow. The other problem, of course, is the light sensors here, right? So, it's not capturing what goes into the eye. If I'm using a device such as a phone in my hand, the light's actually going to be shining away from the sensor. So we know it's going to be particularly crummy for capturing light in the evening, on handheld devices and things like this that we actually care a lot about when it comes to the impacts on the circadian system. The other problem is most of these devices measure and report visual brightness. So what we call photopic illuminance, which is basically a measure of how subjectively bright is light based on its impact on the cone-based visual system. And we know the cone-based visual system is not what dominates the input to the circadian system. What actually matters more is the sort of melanopic input or the non-visual brightness. And that's a brightness that we're actually not consciously aware of. And most devices are not measuring that and not able to measure that. So we worked on developing a sort of novel wearable that would allow us to potentially do this. We wanted a light pin that would be relatively unobtrusive to wear, that could continuously record a whole day of data so that it needs to have sufficient battery life to do that. We needed it to have similar angular responses to the human eye and have a wide dynamic range. We wanted it also to report the entire visible spectrum. So not just to report, some summary variable, but to give us the full visible spectrum using a spectrometer. And from that, we would be able to calculate alpha optical luminances. So these are what are recommended now as sort of the best way of assessing the impact of light on the human circadian system, which is to actually individually establish the impact of light on each of the photoreceptors in the human eye. So each of the 3 types of cones, the rods, and the effect on melanopsin as well. Because we know that the circadian system is going to be some composite of all of those. And to be honest, we don't exactly know what the equation is for that. We know that under certain settings, melanopic illuminance is sort of the dominant, most important factor. But under other settings, it may be more complicated than that. So ideally, you're able to measure and calculate all of them. So we developed this wearable light sensor, and we validated it against a number of sources. I've highlighted Vinitha and Anas here. They were the people who did a lot of the work on this. They're our colleagues at Monash, Malaysia. So we have a lot of back and forth where they're sending us packages, we're testing equipment, we're giving them feedback and so on. And so we're able to show, through calibration, this device could do very well against a number of different light sources for basically establishing the spectrum. And we're using this device, we can capture spectra across the day. And it's really cool looking at these data. It's almost like a spy device. Like you can tell what someone was doing at different times of day. Like you can tell whether they were near a window or whether they were using a particular type of, LED screen across the day in sort of 2-minute snippets. We've just published a paper late last year on this one, if you wanted to look at that in more detail. But yeah, we've done pretty broad geographic coverage of Melbourne at this point in terms of measuring light in people's homes. I definitely made this map before COVID because like just seeing this kind of map is like giving me just anxiety right now looking at it because I assume it's like COVID cases, but it's not. It's many times that we've measured light in. And so you can see, one of the cool things we can do with this device is, we can classify the type of light that someone was exposed to, based on the shape of the spectrum. So here's an example, looking at how bright someone's light was before they went to bed on the x-axis. And then on the y-axis is sort of how relatively bright the light was for the circadian system. And so you can see you sort of get clusters of different types of lights. The incandescent lights, the traditional ones, are sort of down the lower end. The LED and the fluorescent tend to be much brighter. So I think there's lots we can do with this light sensor. And what I'd like to move towards is establishing whether this type of measurement can improve our estimates of circadian timing at the individual level, because that would go towards answering, to what degree is our inaccuracy in phase prediction due to just poor input into the model. So to summarize, I think models based on what we've seen hold actually a lot of promise for accurately predicting circadian phase in real time. The Jewett-Kronauer Forger model continues to be probably the most robust method for predicting circadian timing. We recently did a review of different methods for circadian timing prediction, and this model is by far the most widely tested. It's the only model that's been tested under a very wide number of field and lab conditions, and it continues to perform pretty well. There are other methods that have been established to perform well under one condition and then fall apart under another, such as shift work. But the Jewett-Kronauer Forger model, it loses a little bit of accuracy in shift work settings, as you would expect, but it actually still does just about as well as any other method. So I think it's a really robust model and sort of testament to all the work that went into developing that model in the first place. I think what we're finding now is that light sensitivity parameters are potentially an unexplored avenue for individual differences that we've only sort of just latched onto. And I'm hoping could ultimately enable more accurate individual level predictions. And part of the reason I have that hope is I think it may be easier to measure light sensitivity than it would be to measure someone's intrinsic circadian period. The problem, of course, is if individual differences are largely due to intrinsic period, there's not that much we can do with that right now. We would have to put someone into a forced desynchrony. Or take some sort of alternative method, such as estimating parameters, like a Bayesian type approach where we'd estimate parameters from incoming data over time. But that would require many, many repeated Dilmah assessments in someone if we were going to use that type of method. So it'd be extremely expensive. As I've shown you, we're also working to develop more accurate tools for measuring light exposure patterns, because that's the key input to the human circadian clock, as we all know. So just like to sort of acknowledge a few people at Monash. I've highlighted the people who I've mentioned along the way who have sort of been most involved in all of this. So that's all for now. Thanks very much, everyone. Happy to take any questions.
Great, thanks so much, Andrew. So yeah, it's a Q&A time. Maybe anyone who wants to ask a question, just raise your hand physically or click the emoticon, then I can unmute you.
Can I ask a question?
Yes, please.
Thank you for interesting talk.
Thank you.
And I'm Hyuk-Hong, studying under J.
So nice to meet you.
Yeah, I'm actually currently investigating the sleep-wake cycle as well by using physiologically based model. So I have a
Oh,
couple
excellent.
of,
Okay.
I have a couple of questions. The first one is when we have data missing in data,
Yep.
which is
Yep.
really frequent in real data assessment.
Yep.
So how could you deal with that missing data?
Yeah, that's a really good question. In terms of missing input data, it was something I looked at a bit with like the delayed sleep phase disorder data, because we did have this problem where often there were like gaps in the data where clearly the device was covered. I looked at a bunch of different sort of imputation methods for that, like including really simple sort of single imputation methods like, what if you just fill it in with 0 lux? Or take the raw data compared with, what happens if you fill it in with someone's average level at that time of day,